Okay, guys, let's talk about the last part of this process. We've stepped through the first few stages of the electron transport process. We've seen that bifurcated input through complex 1 and complex 2, feeding that reduced ubiquinone pool, or that dihydroquinone pool, which is then um, uh, moved, the electrons are then moved through that Q cycle, 2 electron to 1 electron adapter process at complex 3, where the ubiquinone is, or the dihydroquinone is reduced, is oxidized back to that aromatic diketone, and the electrons are moved one at a time into that little 100 kD transport protein cytochrome C that surfs along the outer surface of the intermitochondrial membrane. Okay, so now we got to take those electrons out of cytochrome C and move them through the final complex. It's going to pump a bunch more protons for us, and it's also then ultimately going to dump those electrons into molecular oxygen to make water. And there's a bit of a wrinkle to this process because when you really think about it, this is kind of a very dangerous thing that cells are doing. They're playing with fire in terms of making water, and we'll talk about that at the end of this video. Okay, so if we go back over here, again, we're sort of at our final complex, complex 4, right? We've got this reduced cytochrome C, so it's C-type cytochrome covalently bound inside that little 100 kD protein, and we're now going to pump these into this large terminal complex, complex 4. It's about 160 or so kilodaltons, 13 subunits. Again, a fairly big complex. And we're going to repeat a similar theme. We're going to do some 1 electron transfers, and we're going to have some hemes to do it. We're also going to have a new metal complex, right? So the electrons are initially transferred from cytochrome C, not into an iron sulfur cluster, but into a copper center, right? And so the first one is the so-called copper A site. It's up here at the upper outer surface of complex 4, up near the outer, the intermembrane space, the leaflet of the intermitochondrial membrane, near the intermembrane space where cytochrome C is kind of surfing along. And the electrons are first going to flow into this copper center. And this copper center has 2 copper atoms, a couple of cysteines in coordination with it, and then some histidines that help form this sort of copper A site. Those electrons are then going to move into a tetraperol, iron, a heme type tetraperol, but in this case, it's an A-type cytochrome, or an A-type heme, as you're going to see, right? And then they flow on into the business end of this molecule, which is a combination of both another A-type cytochrome, or an A-type heme, and another copper cluster, in this case, the so-called copper B site. And the copper B site is, again, a couple of copper. In this case, it's a single copper atom, right? That's coordinated in a very particular way with some histidines and a nearby tyrosine, right? Okay, so the gives you a little bit more of a look at this. The copper A site, there's, you know, sort of 3-dimensional space. There's those coppers, those nearby cysteines. The A-type heme is, again, the variation. The basic structure is the same, a tetraperol with an iron in the middle. The difference here is that one of those, what would have been a vinyl group in a B-type cytochrome, has this sort of long extended sort of hydrophobic chain hanging off of it. And some of these other groups are different as well, right? But the business end of this molecule, this tetraperol with that coordinated iron, is the same thing. It's just the external substituents of the ring are a little bit different, right? Then we get down to the business end of this molecule, where that A-type heme, there are 2 of these A-type hemes that are near each other, but one of them is forming this sort of essentially the active site, the business end of this molecule, with this single copper atom in the so-called copper B site. All right, so the question now to grapple with is, how does this work? Okay, so because we need to run a bunch of electrons into this thing to make water, we're going to have multiple rounds, multiple cytochrome Cs, right, that are going to feed and kind of activate the copper the copper B site and that A-type heme that's down there, right? So the electrons will come in, they're transferred first into the copper A site, and to get this thing set up, imagine we first have sort of oxidized the iron in the heme, the A-type heme, in the cytochrome, the heme A copper B site, it's oxidized, and that copper is also oxidized. So what you're going to see is that iron in the A-type heme and that copper are going to flip oxidation state. They're going to go between reduced and oxidized, right? Okay, and to get them with their initially oxidized, to get them reduced, we need two cytochromes, right? So two cytochrome Cs come through. We get an electron. It flows downhill all the way through the first A-type heme, through the second A-type heme, and then into that copper, right, into the copper and the copper B site. Second cytochrome C comes through. Now the electrons flow down the same way, but because the copper B site is now, we've reduced the copper from copper 2 to copper 1, the electrons now flow and stop at the A-type heme, reducing it from iron 3 to iron 2. Okay, now we now have reduced iron in that A-type cytochrome and reduced copper. When this happens, that becomes a fantastically attractive binding site for molecular oxygen. And when it binds, the molecular oxygen binds to form a so-called peroxide bridge. That binding and molecular oxygen coordination between those two metals, the oxygen begins to pull the electrons out of the two metals, right? It's not fully oxidized, but this begins to oxidize them, right? They're still coordinated there, but the oxygen has begun to extract those electrons. So these metals begin to move back towards their more oxidized state, copper 2 and iron 3. In that condition, right, what happens, this peroxide bridge that has begun to reoxidize these two metals, that results in absorption, think about it, of abstraction of protons out of the matrix, right? That are sort of, again, these are sort of chemical protons that are pulled up into the complex, and a couple of more cytochrome Cs pass through that pull electrons down, and this results in a rupture of that peroxide bridge and the formation of essentially like a metal-bound oxyhydroxyl group, right? Okay. Two more chemical protons are then pulled in to the complex, creating water, right? And resulting in now the reoxidized iron in that heme A site and the copper B site, right? So by running essentially 4 cytochrome C reduction cycles, right? So we've run 4 molecules of cytochrome C, and we've now pulled in 4 protons. We've taken 1 molecular oxygen and made 2 molecules of water out of it, right? Which is really, kind of amazing that this happens this efficiently and works basically this well. But in addition to getting these 4 chemical protons, right, in this process, what's also true, complex 4 also chemically pumps protons, right? So this is a big bang for the buck. This is kind of like complex 1 gives us a big hit, right? It directly is pumping lots of protons as well as taking up some chemical protons and making dihydroquinone. Complex 4 is doing the same thing. We're taking up chemical protons by virtue of making water out of the matrix, right? But this, and so with that, removes protons, if you want to think about it, from the matrix. But this complex also, as a result of this cycle, physically pumps protons through the structure into the intermembrane space, out of the matrix into the intermembrane space. Now, exactly how that happens, I'm not sure has been completely worked out. This is one of the ideas that's been around for a while, and it has to do with the idea of a sort of a flipping motion of a key aspartic acid residue that's buried essentially in the sort of hydrophobic zone where this water molecule is when it's getting produced, right? And so it's very hydrogen bonded, right? This thing is buried on the interior of the protein, which is not going to be a favorable thing unless you can satisfy those polar groups and keep this thing hydrogen bonded, and that's how it's normally exists in this buried state. But production of water, when the complex 4 does its thing, the water that gets produced essentially displaces that aspartic acid residue, right? And takes its place in the position where it was hydrogen bonding. And that forces this aspartic acid residue essentially to flip out to the intermembrane space, right? And when it does it, the proton that was on that carboxylate group then gets displaced or gets released into the intermembrane space. So we've released a proton. Okay. Now, one of the ideas for what happens now is when the water, when the heme, then, or excuse me, the copper B and the heme A site then get reoxidized. This results in full displacement of the water, which either through a conformational change or something, pushes the aspartic acid residue back down into this buried position where that deprotonated carboxylate group is super unfavorable. Man, that's just not something that this energetically is going to be tolerated for long. And the idea is this results in a tautomerization of a nearby backbone amide group that's attached to this serine, right? So when this thing tautomerizes to form this carbon-nitrogen double bond with turning the carbonyl into a carbon-oxygen bond, this idea is this group was hydrogen bonded to a tyrosine that's near a sort of a proton transfer pathway coming out of the matrix, right? This allows, this tautomerization allows that proton to be abstracted from that tyrosine group to stabilize that group. And when the tyrosine then pulls in another proton, it allows that carbonyl group to then relax essentially back, right, to form the carbonyl group. And we've essentially pulled a proton out of the matrix and then released it by this aspartic acid residue flipping in and out. It's one of the ideas. This has been around for years, and I don't know if this mechanism has held up to the test of time, but it's clearly true that there is something going on, and the elements of this idea that are probably in the ballpark for how complex 4 is also pumping protons actively out of the matrix into the intermembrane space. At the same time, its formation of water is sucking protons out. Okay, so just a quick sidebar. Remember, in addition to the NADH that's being made by TCA cycle, we've got these NADH equivalents from glycolysis that would be really great to run through electron transport. How do we get those in, right? There's no NADH transporter across the outer intermitochondrial membrane, right? So there's sort of two ways in which this generally happens. One is to take dihydroxyacetone phosphate and turn it into glycerol 3-phosphate, right? By taking NADH in the cytoplasm, DHAP, which as you recall is being made by glycolysis, the aldolase reaction, dumping those electrons into DHAP to make glycerol 3-phosphate, which can be transported across the outer mitochondrial membrane. And then there's another enzyme, a dehydrogenase, glycerol 3-phosphate dehydrogenase, that can convert the glycerol 3-phosphate back into DHAP and directly feed through a mechanism not unlike succinate dehydrogenase, the electron transport process through a bound FADH2 molecule. The other big way this happens is one you'll remember, which is to simply take oxaloacetate in the cytoplasm, right? Convert it into malate by running a malate dehydrogenase reaction in reverse, right? To create malate, which can be transported into the mitochondria and then reconverted back through that last step of the TCA cycle into oxaloacetate through the production of NADH that's now localized in the cytoplasm, in the matrix of mitochondria, and can feed complex 1, right? Okay. Now I said something earlier about, cells doing this is kind of like playing with fire, right? Because if you remember something about what's going on here, if we're going to take oxygen and we're going to pump electrons into oxygen one at a time, which is essentially, if you saw how that process worked, that's what happened. When the first two cytochrome Cs came through, we ruptured the peroxide bridge. And when we did that, okay, the oxygen atoms are bound to that metal, but they could come off in principle, right? And if they did, you'd have a radical. You'd have a superoxide radical. At the same time, even if they didn't come off at the superoxide radical, if you didn't get the water into that or protons in there efficiently enough, and you got another electron coming through, you basically are going to form peroxide. All right. These are both a major league problem. Superoxide, that superoxide radical is incredibly reactive. I mean, it will attack almost any carbon-carbon-carbon-nitrogen bond carbon sulfur bond. I mean, it's a super aggressive, right? This is a majorly dangerous molecule to have around. And peroxide itself is an extremely powerful oxidant as well, right? So essentially the process we're running here, electron transport, the terminal stage of this runs the risk of creating extremely dangerous versions, oxidizing versions of oxygen that could do damage to all kinds of biomolecules, including DNA. And oxidative damage to DNA, as we know, can be mutagenic and seriously deleterious, right? Okay, so again, the process of pumping electrons into this complex runs the risk of creating some really dangerous molecules, these superoxide radicals. But interestingly enough, cells, of course, evolution is ready to grapple with that problem. In addition to making this process as efficient as it can within the complex itself to minimize the risk, it's not zero. And the formation of these superoxide and peroxide radicals or peroxides do happen, right? And so there's a backup system. And the backup system consists of a pair of enzymes. One is an enzyme known as superoxide dismutase, which can take superoxide, right, and convert it into oxygen and peroxide. Okay, that's good. I mean, that's really, really good because SOD is a major line of defense for scavenging these super reactive superoxide ions and turning them into something that's less bad. Like molecular oxygen is not so bad. Peroxide, that's not so good, right? Peroxide is still a pretty strong oxidant, right? But there's a second enzyme known as catalase. Catalase will take these peroxides, right? Take two of them and convert them into oxygen and water, right? So the combination of superoxide dismutase and catalase is the backup system for the electron transport chain's tendency, which can't be made zero, to throw off these reactant oxygen species, right? And because this is such a bad problem, if it happens, these enzymes need to be really good. SOD, superoxide dismutase, and catalase need to be really, really good enzymes. So if you had to guess about a kinetic property of those two enzymes in terms of their catalytic efficiency, you could probably guess what their Kcat over Km might be, right? Both catalase and SOD, the primary isoforms of these enzymes that exist in mitochondria to help deal with this problem and in cytoplasm, are extremely efficient enzymes. Their Kcat over Km approaches that theoretical diffusive limit of 10 to the 8th, 10 to the 9th molar per second, per molar per second. So these are essentially catalytically perfect enzymes. And the evolutionary pressure to keep bad stuff from happening from these molecules is driven them to be as efficient an enzyme as is physically possible, right? Okay, last sort of point here, the whole purpose of this, in addition to dumping those electrons, right, so that we can regenerate our oxidizing pools, the other main point of this, as I pointed out before, was to pump protons. All right, so we're taking protons out of the inner mitochondrial, out of the matrix, right, and we're pumping them, as we've seen, across the inner mitochondrial membrane into the outer membrane space. So that's going to result in a separation of both concentration, proton concentration, and charge, all right, across the inner mitochondrial membrane. And that's the energetic condition, the so-called energized mitochondrial condition we're going to use when we see in the next lecture that we're going to use to make ATP. Okay.